Physical mapping is a technique used in molecular biology to determine the relative positions of genes or DNA markers on a chromosome. Unlike genetic mapping, which relies on recombination frequencies, physical mapping uses direct measurements of DNA distance. Key methods of physical mapping. Restriction enzyme, mapping. Enzymes that cut DNA at specific sequences known as restriction sites are used to create fragments. The fragments are then separated by size using gel electrophoresis. The order of fragments can be determined by analyzing their overlap. Cloning and sequencing. DNA fragments are cloned into vectors and sequenced. The sequence information is used to identify genes and their relative positions. Fluorescence in situ. Hybridization, or FISH, involves the use of fluorescent probes that bind to specific DNA sequences. These probes allow us to visualize their location on chromosomes. This technique can be incredibly useful for identifying genes or detecting chromosomal abnormalities. There are several important applications of physical mapping. First, gene identification. Physical mapping can help identify genes within a region of interest. Then, there's the study of chromosomal rearrangements. It can be used to detect and study abnormalities such as deletions, duplications, and inversions. Another critical application is genome assembly. Physical mapping is essential for assembling large genomes from smaller DNA fragments. Lastly, comparative genomics. This technique can be used to compare the genomes of different species and identify regions of similarity and difference. Of course, there are some challenges and limitations associated with physical mapping. For instance, large genomes can be particularly challenging due to the difficulty in handling and analyzing large DNA fragments. Repetitive sequences also pose a problem, as they can make it difficult to accurately determine the order of fragments. Additionally, physical mapping can be time-consuming and expensive, especially when dealing with large genomes. In summary, physical mapping is a powerful tool for understanding the structure and organization of genomes. By providing precise information about the location of genes and other DNA elements, it plays a crucial role in various areas of molecular biology and genetics.